Now I'm talking about electron transport system, oxidative phosphorylation. The respiratory breakdown of glucose in presence of oxygen is an oxidative process. During this process, several intermediates such as phosphoglyceraldehyde, pyruvic acid, isocitric acid, alpha-ketoglutarate acid, succinic acid, and malic acid are oxidized. Each oxidation step involves release of 2H, which goes to reduce various enzymes with NAD plus and FAD. The pair of hydrogen is usually picked from the substrate by NAD plus or FAD. Synthesis of ATP during oxidation of coenzyme in ETS of aerobic respiration is called oxidative phosphorylation. In this, substrate is first oxidized by releasing a pair of hydrogen atom with the help of dehydrogenase enzyme. This hydrogen atom becomes ionized into protein and electrons. They are picked up by NAD plus and is reduced to NADH plus H plus. Reduced NADH plus H plus transfer one proton and two electron to FAD, and it is reduced to FADH2, getting one more proton from the medium. FADH2 transfers two electron to ubiquinone UQ through FeS iron sulfur protein, causing reduction of UQ to UQH2 in the perimitochondrial space. Ubiquinone is also known as coenzyme Q or COQ. It is a mobile carrier and no bound to stocked particle F1. The name ubiquinone is given due to its ubiquinous nature. It is a highly mobile carrier between flavoprotein and cytochrome. The proton after reduction of UQ do not participate in the oxidation reduction process, and only electron participate in the oxidation and reduction of cytochromes. These electrons after passing through cytochrome finally reach to O2. The penultimate electron acceptor is cytochrome A3. Cytochrome A3 contain Fe and Cu both, whereas all other cytochrome carriers contain iron. The cytochrome A passes electron to Fe of cytochrome A3, and copper of cytochrome A3 help in transferring electron to O2. The transfer of electron takes place from higher redox potential E0 to low redox potential. NADH is oxidized at E0 of minus 0.32 V, and O2 is reduced at plus 0.82 V. During this journey, electrons lose some of their energy at certain points, which help in the formation of ATP with the help of ADP and inorganic phosphate. Oxidation of one NADH plus H plus molecule yields 3 ATP molecules as under. Between NAD and FAT equal 1 ATP, 12.2 kilocalorie. Second, between cytochrome B and cytochrome C, 1 ATP, 15.2 kilocalorie. Third, between cytochrome A and cytochrome A3, 1 ATP, 24.5 kilocalorie. Net gain of ATP molecules during glycolysis, 6 ATP. In prokaryote, total net gain of ATP molecule produced by the oxidative breakdown of glucose is 8 plus 30, 38 ATP. In eukaryote, the net gain of ATP is lesser because 2 ATP molecules are consumed in transferring NADH plus H plus from cytosol to mitochondria. These two molecules of NADH plus H plus are generated during glycolysis in reaction. In eukaryotic, net gain of ATP molecules 38 ATP minus 2 ATP equal to 36 ATP. So we can say the electron transport chain or respiratory chain is a series of electron carriers in the membrane of the mitochondria. It is present in the inner mitochondrial membrane. Through a series of reactions, the high energy electrons are passed to oxygen. In the process, a gradient is formed and ultimately ATP is produced. Electron transport requires oxygen directly. Electron transport takes place on cristae of mitochondria. Electrons are transferred through cytochromes, cytochrome B, cytochrome C1, cytochrome C2, cytochrome A1 and cytochrome A3, and finally reaches molecular oxygen. Enzyme useful for terminal oxidation is cytochrome oxidase or terminal oxidase. Both cytochrome A and A3 form a system called cytochrome oxidase. Copper is also present in cytochrome A3. In addition to iron, synthesis of ATP during oxidation is called oxidative phosphorylation. 3 ATP and 1 H2O are formed by the oxidation of a molecule of reduced coenzyme first or second between NADH2 and flavoprotein, between cytochrome B and cytochrome C1, between cytochrome A and A3. Chain level phosphorylation takes place in oxysomes. In the respiratory process, release and utilize the energy stored in NADH plus H plus and FADH2. This is accomplished when they are oxidized through the electron transport system and the electrons are passed on to oxygen, resulting in the formation of water. Chemo osmotic mechanism of ATP synthesis has been postulated by Peter Michel in 1961. Electron from NADH produced in the mitochondrial matrix during citric acid cycle are oxidized and NADH dehydrogenase complex 1, and electrons are then transferred to ubiquinone located within the inner membrane. Ubiquinone also receives reducing equivalents via FADH2 complex 2 that is generated during oxidation of succinate in the citric acid cycle. The reduced ubiquinone, that is ubiquinol, is then oxidized with the transfer of electron to cytochrome C via cytochrome BC1 complex 3. Cytochrome C is a small protein attached to the outer surface of the inner membrane and acts as a mobile carrier for transfer of electrons between complex 3rd and 4th. Complex 4th refers to cytochrome C oxidase complex containing cytochrome A and A3 and 2 copper center. When this electron passes from one carrier to other via complex 1 to 4th in the electron transport chain, they are coupled to ATP synthetase, that is complex 5th, for the production of ATP from ADP and inorganic phosphate. The number of ATP molecules synthesized depend on the nature of electron donor. Oxidation of one molecule of NADH gives rise to 3 molecules of ATP, while that of one molecule of FADH2 produces 2 molecules of ATP. The number of ATP molecules synthesized depend on the nature of electron donor. Oxidation of one molecule of NADH gives rise to 3 molecules of ATP, while that of one molecule of FADH2 produces 2 molecules of ATP. Although the aerobic process of respiration takes place only in the presence of oxygen, the role of oxygen is limited to the terminal stage of this process. Yet the presence of oxygen is vital, since it drives the whole process by removing hydrogen from the system. Oxygen acts as a final hydrogen acceptor. Unlike photophosphorylation, where it is the light energy that is utilized for the production of proton gradient required for photosynthesis, for phosphorylation, in respiration, it is the energy of oxidation reduction utilized for the same process. It is for this reason that the process is called oxidative phosphorylation. ATP synthetase, that is complex 5th, consists of 2 major components, F1 and F0. The F1 headpiece is a peripheral membrane protein complex and contains the site for synthesis of ATP from ADP and inorganic phosphate. F0 is an integral membrane protein complex that forms the channel through which protons cross the inner membrane. The passage of proton through the channel is coupled to the catalytic site of the F1 component for the production of ATP. For each ATP produced, 2H plus passes through F0 from the inner membrane space to the matrix down the electrochemical proton gradient. It is possible to make calculation of the net gain of ATP for every glucose molecule oxidized. But in reality, this can remain only a theoretical exercise. These calculations can be made only the certain assumptions that there is a sequential orderly pathway functioning with one substrate from the next, and with glycolysis, TCA cycle and ETS pathway following one after another. The NADH synthesized in glycolysis is transferred into the mitochondria and undergo oxidative phosphorylation. None of the intermediate in the pathway are utilized to synthesize any other compound. Only glucose is being respired. No other alternative substrate are entering in the pathway at any of the intermediary stages. Number of ATP formed by the oxidation of glucose during glycolysis is 10. Substrate level phosphorylation equal to 4. Chain level phosphorylation equal to 6. Net gain of ATP through glycolysis is 8 when glucose is completely oxidized. Number of ATP formed for the complete oxidation of each molecule of pyruvic acid is 15. Therefore, for 2 molecule of pyruvic acid is 30. Number of ATP formed by complete oxidation of a molecule of glucose, that is 180 gram, is 40. Number of ATP utilized during respiration is 2. Net gain of ATP during aerobic respiration is 38. But this kind of assumption are not really valid in a living system. All pathway work simultaneously and do not take place one after other. Substrate enter the pathway and are withdrawn from it as when necessary. ATP is utilized as and when needed. Enzymatic rates are controlled by multiple means. Hence, there can be net gain of 36 ATP molecule during aerobic respiration of one molecule of glucose.